part 1. First and foremost thing is that one should know about the constituents of animal house facility. There are 5 main constituents: physical plant, environment, animal care, personnel, and equipments or SOPs. In this lecture, we will discuss about the physical plant and environment. There are 2 types of animal colonies: conventional type and barrier type. Conventional type means an animal carrying the full or undefined burden of organisms usually associated with its species. And barrier type is again divided into 3. The specific pathogen-free SPF colony means free from specific pathogens and parasites. Notobiotic animals, notos is a Greek word which means well known, and biota means all life. So an animal or a system in which all the life forms are known is called as notobiotic animal. Germ-free animals, that is animals reared in the absence of microbes. Clean and conventional colony is acceptable for most of the research and testing provided these are free from common diseases. Now we come to physical plant. The animal environment, housing and management practices are essential for animal well-being, quality of the research programs, and health and safety of the staff. The physical facilities include corridors, animal rooms, doors, walls, floors and ceilings. Utilities include water lines, drain pipes, electrical connections. Electrical connections should be accessible through service panels or shafts in corridors outside the animal rooms. The storage areas, separate rooms or adequate space should be available for feed storage, bedding material storage. Bedding and feed should be stored in separate area. Equipment storage room should be there. Animal waste disposal area should be there. Then facilities for sanitization include a dedicated central area for sanitizing cases and equipment should be there. There should be ease of access to the wash area. There should be provision for safe bedding disposal and free washing activities. The experimental area. The experimental rooms should be adequate, appropriate and suitable for experimentation. It should have minimum infrastructure in the room. Suitable equipment and apparatus involved with the experimental procedure should be available. It is recommended that experimental room should be away from the holding or breeding rooms. Then housing. Lab animals are very sensitive to their living conditions. Cages are used for accommodating them. The cages provide security, accommodation. They should be economic. There are different types of cages like holding cages, metabolic cages, activity cages, breeding cages, and squeeze cages. Then bedding material should be easily available. It should be cost effective, moisture absorbent, and non-toxic. There are different types of bedding materials like paddy husk, corn cob, paddy straw, paper shred. Then caging or housing system. It should provide a secure environment that doesn't allow escape of or accidental entrapment of animals or their appendages between opposing surfaces. Cages should be free of sharp edges or projections that could cause injury to the animals. The type of cages, one should know about the type of cages and whether it is appropriate for the species. The recommended space allocations for common laboratory animals is given in CPCSEA guidelines. CPCSEA stands for Committee for the Purpose of Control and Supervision of Experiments on Animals. Recently, this CPCSEA, the name is changed to CCSEA, Committee for the Control and Supervision of Experiments on Animals. P is removed. These are different types of cases. These are the pics of different types of cases for accommodating laboratory animals. Then conventional type of housing. In conventional type of housing, the cages should be mounted on clean racks. The floor area per animal requirement is different for different species. For rats, mouse, guinea pig, rabbits, these are different, and the cage dimensions are also different for them. These are some of the pics for barrier type of housing used in case of germ-free, for accommodating germ-free animals. Then this is the photo showing production of rabies vaccine, inoculated rabies in different stages of paralysis. This photo is from Partial Institute, Kasauli, India. These are some of the pigs for accommodating this. These are the cages for accommodating guinea pigs, rats and mice cases. and these are rabbit cases. Automatic case washer is available in the market for washing the cases automatically. Water washer for washing water bottles automatically are also available. Then bedding automation is also there. The bedding is put inside the laboratory animals cases automatically by using machines. There are some advanced type of cases used for research purpose. This is dynamic containment case. In the field of toxicology and pharmaceutical research, one of the main issues is the protection against volatile chemical agents and contaminated rodents. It protects the operator even when the case is partially open. One can assess the animal for inspection or food refilling, while the powerful aerial barrier prevents any compound or metabolite escaping from the case. This is an ISO-case type of housing. The ISO-case system consists of a dedicated case, a dedicated rack and air handling unit, and a dedicated class 2 biosafety station. The individual cases are hermetically sealed and have their own filtered positive airflow. The ISO case is capable of working at a highly consistent negative pressure, and it is able to maintain the pressure mode when disconnected from the ventilating rack. By using strict sterile techniques, the mice or lab animals are kept germ-free in this casing system. It also requires less space and reduces animal house cost. This is green line IBC casing system. IBC stands for individually ventilated case. It is used to keep an animal separated from other animals and possible exposures, including exposure by air. DCM stands for Digital Ready Green Line Mouse Rack. It is new revolutionary mouse rack, fully compatible with current and future technologies. The benefits are improved case density, offers up to 107 cases per square meter. Exceptional filtration protects both inhabitants and facility staff. It saves as much as 25% of time in case changing. Increased visibility is there, reduced weight due to half-weight wire bar. Constant environmental conditions mean better science. This is double-decker IBC casing system. The double-decker provides real environmental complexity in IBC casing. Climbing, playing, and exploration are the key elements of normal rat behavior, along with the ability to exhibit bipedal posture. Unlike traditionally barren cases, the unique double-decker provides real usable three-dimensional space. Sections of the case extend to a height of 37 cm, giving rats ample room to stretch. It provides easy handling and space saving. Easy observation is there. Case locking system is present. Enhanced containment and bio-exclusion. Seal and filter is present. Top slim line. It is an air handling unit that is fitted with a microprocessor controlled touch screen, which controls the speed of both supply and exhaust blowers while displaying the temperature and related humidity. Total control is at your fingertips. The second constituent is environment. The environment of animal room, macro environment, and the animal cases, that is the micro environment, is an important factor on which the production and experimental efficiency of the animal depends. Animals are very sensitive to environmental changes, hence sharp fluctuation in temperature, humidity, light, sound, and ventilation should be avoided. The constant and comfortable environment is required to ensure the health of experimental animals as well as personnel working with them. This is the pic showing the microenvironment immediately surrounding the animal, and macroenvironment means the environment of the room in which the animals are kept. The microenvironment or primary enclosure is the physical environment immediately surrounding the animal, cage, pen or stall. This enclosure should allow for normal physiological and behavioral needs of the animal with easy access to food and water and adequate ventilation. The primary enclosure should allow the animal to remain dry and clean, provide a secure environment free from sharp edges and that might lead to injury and kept in good condition. It depends on the density of animals, that is number of animals in the case, production of ammonia. High relative humidity enhances the production of ammonia, bedding material, soiled or dry, condition of the case, intact or cracked. The macro environment or secondary enclosure may be described as the physical environment of the room, barn or outdoor habitat of the research animals. This should include the construction, temperature, humidity, ventilation and lighting. The microenvironment is most easily assessed when you walk into the room. Does it feel hot, cold, humid? Does it have odors? Is the animal room too bright or dark? Temperature. Heat is the most obvious and important factor which affects the living organism. A constant room temperature is essential. Variation in room temperature causes changes in food and water intake. The heat production and heat loss should have a degree of control as the body of the animal is concealed with fur and not able to sweat and respond to the changes in ambient temperature by altering the metabolic rate. Temperature and humidity control unit should be properly installed outside the animal rooms and the ducts should be properly placed inside the room. Air conditioning is an effective means of regulating the temperature and humidity. The comfortable temperature recommended is 22 plus minus 2 degrees centigrade. A change in temperature of 4 degrees centigrade can cause tenfold alteration in biological response. And lighting can affect the physiology, morphology and behavior of various animals. Lighting should be diffused throughout an animal room to provide sufficient illumination for the well-being of animals, or good housekeeping practices, adequate inspection of animals, safe working conditions. The recommended lighting is 12 hours dark and 12 hours light. The intensity of light recommended is 300 to 400 lux. And ventilation, sufficient fresh air supply or oxygen and effective removal of respiratory odors is very essential. 10 to 15 fresh air room changes per hour is recommended. Either 100% fresh air or recirculated air should be there. Supply and exhaust measured with anemometer. Odors in animal facility results from bacterial decomposition of the excreta. The most common air contaminant is ammonia. The relative humidity recommended for laboratory animals will vary from 50 to 60%, ranges in between 30 to 70%. High relative humidity enhances the production of ammonia in animal cases due to decomposition of excreta of animals, which can cause pathological changes and respiratory disorders in animals. The viability of airborne microorganisms seems to be lowest at a relative humidity of 50%. Noise. Intense, unexpected sounds seems to be more harmful than constant noise. Loud noises precipitate epileptic seizures in several species. Intermittent noise may also affect the breeding performance. The recommended noise level is 50 decibels. Space recommendations. The space needs of animals are complex. Overcrowding of animals can occur easily and cages should be evaluated for number of animals. These are the housing and environmental requirements for different species: mouse, rat, hamster, guinea pig. The weight of mouse should be 25 to 30 gram. Rat weight is 200 to 300 gram. Hamster 80 to 100 gram. Guinea pig 400 to 500 gram. Type of housing is caged for all species. In guinea pig, pen or also pen can be used. Floor area per animal requirement is 65 to 100 square centimeter for mouse, 100 to 150 square centimeter for rat, 90 to 120 square centimeter for hamster, 300 to 600 square centimeter for guinea pig. Cage height should be 12 centimeter for mouse and hamster, 14 centimeter for rat, 18 centimeter in case of guinea pig. Room temperature should be 22 to 24 degrees centigrade for all species. Relative humidity should be 45 to 60% in case of mouse, hamster and guinea pig, and it should be 50 to 60% in case of rats. Ventilation, 10 to 12 air changes per hour is recommended for all species. Light intensity should be 300 to 400 lux. Photocycle, 12 hours light and 12 hours dark is recommended for all species. The housing and environmental requirements for rabbit, cat, dogs and primates. The rabbit weight is 2 to 2.5 kg, cat 3 to 5 kg, dog's weight is 12 to 15 kg, and primates 3.5 to 8 kg. Type of housing is cage. In case of rabbit, cat and primates, in dog run area is provided. Floor area per animal 3700 to 4600 square centimeter in case of rabbit, and in cat 2500 to 3500 square centimeter. In dog 7000 to 12000 square centimeter. Primates 4000 to 6000 square centimeter floor area per animal is required. Cage height should be 36 centimeter in case of rabbit and cat. In primates 72 centimeter. Room temperature 22 to 24 degrees centigrade for rabbit, others is air-dried. Relative humidity 45 to 60% in case of rabbit, dogs, primates, and it is 50 to 60% in case of cat. Ventilation 10 to 12 air changes per hour for rabbit, and it is continuous in other species. Light intensity same 300 to 400 lux, and photocycle light is to dark period is 12 hours. Then sheltered or outdoor housing. Social interaction is very important for higher animals like monkeys and dogs. Outdoor housing or run area is essential for social animals like monkeys and dogs. The run area is properly built and escape proof. The material used for construction should be corrosion proof and provide proper protection from extremes in temperature. This lecture will be continued on 22nd of February. The rest of the students of animal house facility like equipments and SOPs, personnel, animal care will be discussed in that lecture. In last class, some students asked about the transgenic animals in detail. The transgenic animals, we all know these are the animals, most commonly mice, that have had a foreign gene deliberately inserted into their genomes. Such animals are most commonly created by microinjection of DNA into the pronuclei of a fertilized egg, which is subsequently implanted into the oviduct of the pseudo-pregnant surrogate mother. And this results in the recipient animal giving birth to genetically modified offspring, and then the progeny is then bred with other transgenic offspring to produce a transgenic line. This photo shows two transgenic mice positioned either side of playing mouse. The transgenic mice have been genetically modified so that they carry a green fluorescent protein which glows green under blue light. The importance is the transgenic animals are routinely used as models in biomedical research to study obesity, heart disease, diabetes, arthritis, anxiety, aging, Alzheimer's, and Parkinson's disease. Over 95% of those used are genetically modified rodents, predominantly mice. These are important tools for researching human disease, being used to understand gene function in the context of disease susceptibility, progression, and to determine responses to a therapeutic intervention. The mice have been genetically modified to naturally produce human antibodies for use as therapeutics. There are several ways of creating the transgenic animals, several ways of inserting the DNA into the genome of mouse. These approaches are, first is retroviral approach. It is not routinely used. Second is standard transgene approach, and third is called as gene targeted transgene approach. In case of retroviral, by using retroviral infection, in the donor female mice is taken, the mouse embryo at 8-cell stage, the retrovirus containing the transgene is inserted into the embryo, and it is then implanted into the pseudo-pregnant mother, surrogate mother, and then the offspring are screened for the presence of transgene. In case of a standard transgene approach, the donor female mice is taken, DNA injected into the male pronucleus of fertilized oocyte. Then they are cultured in vitro, and the early mouse embryos are implanted into the pseudo-pregnant mice, and the offspring produced are screened for the presence of transgene. In case of gene-targeted standard transgene approach, the embryonic stem cells are taken. DNA with targeted transgene is inserted into the embryonic stem cells. These embryonic stem cells, targeted embryonic stem cells are selected and expanded, and then the targeted embryonic stem cells are injected into early mouse embryo, and then the embryo is implanted into the pseudo-pregnant mice. These embryonic stem cells and the donor blastocyst, they derive from mouse lines having different coat colors. So after successful implantation of embryo in these pseudo-pregnant mice, the offspring produced is chimeric offspring with variegated coat color. Then this is again mated with wild type of mice to check the germline transmission of transgene. And after this cross, heterozygous progeny is produced, certain percentage of heterozygous progeny is produced carrying the transgene. Then finally intercross between these heterozygotes results in homozygous mutant mice, and this at an expected 25% of frequency unless the mutation is detrimental to embryo survival and development. And then various experiments are carried out to on this homozygous mutant mice to test the functionality of this genetic modification. This is how the transgenic animals are created. These are the references for making this presentation.